So as you can see in the photographs of the slides, the animal slide is on top, and then the bottom set of slides is the plant cells, and then the illustration in the middle help you kind of understand what you're supposed to be seeing. So in early prophase, you can start to see these structures are called the centrosomes, and the two barrel-shaped structures are the centrioles. So those centrosomes are going to start moving to opposite ends of the cell. The nuclear envelope will begin to disappear. The nucleolus will disappear. Chromatin is beginning to condense and form into chromosomes. And you can see that happening even more in prophase. The centrosomes are moving to opposite ends of the cell. You can see spindle fibers forming. The spindle fibers are microtubules, and they're sometimes called kinetochore fibers because they will attach to the kinetochores of each of the chromosomes. You can see that the nuclear envelope is almost gone. These little purple lines represent what's left. The nucleolus has disappeared, and the chromosomes are visible under the microscope. You can see them here in the animal cell, and you can see them in the plant cell. Then you move to prometaphase, and the spindle fibers have attached to the kinetochores. In metaphase, the chromosomes have lined up on the metaphase plate. In anaphase, the sister chromatids are separating. And then in telophase, all of the events that happened in prophase are going to be reversed. So you will start to see nuclear envelopes reforming. The nucleolus will reappear. The chromosomes will diffuse back into chromatin. The centrosomes with the spindle fibers will also disperse, won't be visible anymore. You'll see the centrioles in an animal cell, but you won't in a plant cell. In cytokinesis, in the animal cell, you'll see the formation of a cleavage furrow here, and then the two cells will split. And in a plant cell, you'll see the formation of a cell plate, which will eventually form into a cell wall. And then this is basically the notes of every phase. In early prophase, centrosomes have duplicated, so you have two centrosomes. Chromatin is condensing into chromosomes. Nuclear envelope is fragmenting. By prophase, the nucleolus has disappeared. Duplicated chromosomes are visible. You can see them as darkly stained rod-shaped structures in the animal and the plant cell. The centrosomes begin moving apart, and the spindle is in the process of forming. In prometaphase, the kinetochores are attached to the spindle fibers are attached to the kinetochores. And in metaphase, the chromosomes line up at the metaphase plate or the center of the spindle. In anaphase, the sister chromatids pull apart. And in telophase, daughter cells are forming. As nuclear envelopes, nucleoli reappear, chromosomes will diffuse back into chromatin again. All right, cytokinesis in plant and animal cells is a little different. Cytokinesis is the division of the cytoplasm. So it's the point at which the parent cell splits into two daughter cells. In animal cells, cytokinesis begins with the formation of a cleavage furrow. And in plant cells, it begins with the formation of a cell plate. And the cell plate is formed by many small membrane-bounded vesicles that are made by the Golgi apparatus. They will fuse together to form the cell plate. So this is an image of cytokinesis in animal cells, and then cytokinesis in plant cells. All right, functions of mitosis. Functions of mitosis are that it permits growth and repair. In mammals, mitosis is necessary when a fertilized egg becomes an embryo. The fertilized egg, called the zygote, must divide many, many, many times, and that division is called mitosis. That cell division or nuclear division of those cells is called mitosis. When the embryo grows into a fetus, when the child grows into an adult, during all those times, cells are having to divide so that the human can get larger. And also during repair, when a cut heals or a broken bone mends, new cells have to be produced to help the individual heal. All right, so stem cells are cells that retain the ability to divide after the organism, has grown into an adult. And there are two types of cloning which use stem cells. There's therapeutic cloning, which is used to produce human tissues. Therapeutic cloning can occur from adult stem cells or embryonic stem cells. And then reproductive cloning must use embryonic stem cells. Reproductive cloning is the production of a new individual. We've done that with some mammals, but of course not humans at this time. This shows you how cloning works. You have to take stem cells, which are stem cells, well, I'm sorry, I apologize. I didn't mean to say stem cells. You have to use cells from the animal that you're cloning. The cells have to be in stage G0. That means they're not dividing cells. They're not going to continue to divide. You have to take and remove the nucleus out of that G0 cell. Then you take the nucleus from the egg cell and you discard it. So you fuse the G0 nucleus into the egg cell. So there's an egg cell that doesn't have its own nucleus. It now has the nucleus from these cells that are in stage G0. And then they are allowed to grow in a culture medium. They are implanted. The embryo that is formed in this culture petri dish is implanted into a surrogate mother, and the clone is born. That's reproductive cloning. Then you can have therapeutic cloning, which uses the same process. You take the nucleus out of a G0 somatic cell, and you place it into the egg, fuse it with the egg that doesn't have its nucleus anymore. You culture or grow these cells, and then you can grow individual tissues like nervous tissue, blood, and muscle. Skin is another one. All right, so we're going to talk about cancer in section 4. The term benign refers to a tumor that is not cancerous. It's got a capsule around it. It's not going to invade other tissues. It's not going to spread. But if we call it a malignant tumor, then the person is diagnosed with cancer. So malignant tumors are cancerous and they can spread. Now, cancer cells are different from normal cells. They lack specialization or differentiation. They're not specialized to do anything. They just keep dividing. That's all. That's what they do. It says here they're immortal. They can enter the cell cycle repeatedly. So really cancer is mitosis that's uncontrolled. Okay. The nuclei of cancerous cells is abnormal in some way. And cancerous cells, even though they're abnormal, don't undergo apoptosis. They're not programmed to die. And that's what makes it cancer, and that's what makes it dangerous. So the way that tumors form is the fact that cancer cells have lost this property called contact inhibition. Normally, when you have tissues forming, cells grow, and then when they start to contact each other, they stop dividing. That's contact inhibition. But cancerous cells have lost that. So even though they're in contact with other cells like themselves, they still continue to grow. Cancerous cells undergo metastasis, which means that the tumor can break apart and it can spread to other organs. Another thing that occurs with cancerous cells and tumors is that this process called angiogenesis, the formation of new blood vessels. And so the tumor will receive nutrients and oxygen, sometimes even when the person's healthy cells won't get enough, nutrients and oxygen, won't get enough of a blood supply because it's now being diverted to the tumor. This is an illustration of the progression of cancer, starting with a cancerous cell, and then the growth of the tumor. One of the cells now is going to spread either into the lymphatic tissue or the blood vessel. And because of that, because these cells can spread through the lymphatic tissue and the blood, you can find tumors a far distance away from the original tumor. So a table that compares cancer cells to normal cells. Cancer cells are not differentiated, whereas normal cells are. Cancer cells have abnormal nuclei. They don't undergo apoptosis when something is wrong. Excuse me. Cancerous cells don't have contact inhibition. Cancerous cells are disorganized and multi-layered, whereas normal cells tend to form one organized layer of cells. And cancer cells can spread. They undergo metastasis, whereas normal cells remain in their original tissue. They don't spread to other tissues. So, how does cancer originate? How does it start? It starts with oncogenes and tumor suppressor genes. So, oncogenes are formed from mutated proto-oncogenes. So a proto-oncogene is a good gene. Proto-oncogene codes for proteins which promote the cell cycle in various ways, and a mutated proto-oncogene can become an oncogene, which is a cancer gene. Okay? So proto-oncogenes promote the cell cycle under normal conditions, healthy conditions, but if they're mutated and become an oncogene, then they cause the cell cycle to go out of control. Tumor suppressor genes code for proteins which inhibit the cell cycle and promote apoptosis. And they're good genes because they inhibit the cell cycle in abnormal cells. But if a tumor suppressor gene becomes inactive, cancer can develop. So cancer can develop from a mutated inactive tumor suppressor gene, or it can develop from mutated proto-oncogenes which have become oncogenes. And here you can see different causes that will mutate, that will cause the proto-oncogenes to become mutated or the tumor suppressor genes to become mutated. It can be caused by heredity, genetic factors, pesticides and herbicides, viruses, and radiation. A growth factor activates signaling proteins in a stimulatory pathway that extends to the nucleus. So a growth factor will bind to a receptor protein in the plasma membrane, and it will activate a signaling protein, and that will stimulate the cell cycle. So in a proto-oncogene which codes for growth factors, if it becomes an oncogene, it can lead to like uncontrolled growth. And then a tumor suppressor gene codes for a signaling protein in an inhibitory pathway. But if it's mutated, then it will not suppress cell division, and it will lead to uncontrolled cell growth because it's not suppressing it. So just basically, proto-oncogenes stimulate cell division. When they become cancerous, cell division is not controlled anymore. And it says that here, mutated proto-oncogenes lead to uncontrolled cell division. There are 100 identified oncogenes or cancer genes, which can lead to tumors, One is called BRCA1 or BRCA1. Mutations of this gene can cause breast cancer and ovarian